Hello, this is Mrs. Kasprowitz, and I am going to go over with you the Science Practices Review, which are multiple choice questions from previous exams, similar to the ones that you might see on your test on Monday. So let's take a look at some of these. Question number 1 says, base your answer to the question on the information below and on your knowledge of biology. A student designed an experiment to determine if air temperature has an effect on the rate of photosynthesis in corn plants. So again, we've got a problem here, and our problem is going to tell us what we're changing, which is our independent variable, and what we're measuring, which is our dependent variable. So in this case, they're asking us to identify the independent variable, which is what we're changing. And in this case, we're changing the air temperature at which the plants are going to be grown. So the answer to this question is going to be number 1. So we design an experiment to determine if air temperature, that's the thing we're changing, has an effect on rate of photosynthesis, which is what we're going to measure. So the independent variable is the air temperature. Number 2, students noticed that some of their classmates had a hard time concentrating during class. They thought it might have some connection with the fact that these students consume energy drinks just before class. An experiment was proposed to find out if there's a connection between energy drinks and the lack of the ability to concentrate in class. A properly designed experiment to determine this would include. All right, so we're going to do a test to see if drinking energy drinks before we go to class is going to have us have difficulty paying attention in class. So the whole class drinks an energy drink and no water at all for the entire time of the experiment. So would that be a properly designed experiment? No, we don't have a control group. The whole class drinks water and no energy drinks at all for the entire time. So again, we need to have two groups here going, one that's going to be our control and one that's going to be our experimental. And here we've only got one group, which is the whole class. The students drink both water and an energy drink just before class. Well, no, we don't want them to drink both because then we're not going to be able to tell the difference. And again, we're dealing with all the students and not breaking them up into groups. So number 4, half the students drink water and the other half drink an energy drink just before class. So this would be a proper experimental design because we have a control group, water, and we have the energy drink as our experimental group, so we can then compare the results, all right, between the two groups. So best answer there is number 4. Sorry about that. All right, next one. Number 3. So a student collected data from an experiment on muscle fatigue. In order to interpret these data, the student should ignore the data because they don't support the hypothesis, recalculate the data so that the numbers are easier to work with, share the data with a student who has none, or organize the data into a table or graph. So if we're going to interpret the data, we need to first organize it. So that's going to be number 4, organize the data into a table or graph. Number 4, a student performed an experiment to see if water temperature affects the level of activity in aquatic snails. The student set up 4 tanks with 5 snails in each tank. All 4 setups were identical in every way except for the temperature of the water. In order to make the conclusions more valid, the student could. All right, so let's take a look and see what we're changing and what we're measuring and what's going on here. So we're going to change the water temperature. That would be our independent variable. And we want to see how it affects the level of activity. So that would be our dependent variable. All right, we set up 4 tanks. We've got 5 snails. They were all identical in every way except that the temperature of the water changed in the tanks. Okay, so everything else was the same. So we've got all these constants and the one variable was the water temperature. All right, so in order to make the conclusions more valid, we are not going to want to change the pH because that would add a variable. We're not going to want to change the size of the tank. That would add a variable. we're not going to want to carry out the experiment for a shorter period of time. So the best answer here is number 4. We could always improve our experiment, or the other way to say that, make the conclusions more valid by using more snails, increasing our sample size or increasing the number of test subjects. Number 5, a student wondered if butterflies would show any differences in their wing color if, as caterpillars, they were grown in the dark or grown in bright white light. Which statement would be a possible hypothesis for this experiment? So what are we changing for the butterflies? That's going to be our independent variable, which is going to be, are they grown in the dark or are they grown in the light? So the amount of light is going to be our independent variable. We're going to measure or observe, in this case, the wing color. All right. So a hypothesis has to have both your independent and your dependent variable to be a hypothesis. So caterpillars exposed to bright white light will have more blue and green in their wings when they become butterflies than caterpillars kept in the dark. Maybe. Will caterpillars kept in the dark have brighter wings when they become butterflies than caterpillars exposed to bright white light? Big clue here that this is not a hypothesis is that there's a question mark here. So that's a no. Number 3, 10 caterpillars are kept in the dark, 10 are kept in the light, and then they're allowed to develop into butterflies. That is the experimental design, not a hypothesis. Results show that caterpillars kept in the dark and exposed to bright light had the same wing color when they became butterflies. So this would be the analysis part or the results part. So the hypothesis goes back to number 1. We're going to expose them to white light, and we expect them to have different colored wings than those that were carried out in the dark. So that's our hypothesis. Number 6, in order to be accepted, a scientific theory must be widely tested and supported by extensive data, based on the results of a single experiment, controversial and cause debate, or in line with all previous historical ideas. So remember, a scientific theory is where we have an explanation for a phenomenon that we've tested many, many times, and we've had many of the same observations. And so now that's supported by lots of data. So that's going to be number 1. Number 7, reasons for conducting peer review, the report repeat part of the scientific method, include all of the following except. So we want to pick the one that does not apply to peer review or to repeating our experiment and reporting our results. Analyzing the experimental design. So do our peers take a look at how we design the experiment? Yes, they do. All right. Do our peers point out possible bias? Yes, they do. Do the peers that take a look at our experiment identify an illogical conclusion? Yes, they do. Do they change the data to support the hypothesis? No, they don't. So that would be the exception. So number 4. Number 8, the current knowledge concerning cells is the result of many investigations and observations by scientists. The work of these scientists forms a well-accepted body of knowledge about cells. This body of knowledge is an example of, well, it's not a hypothesis. We're not making a prediction. It's not an experiment that they just described. It's not a research plan, so it's not like a procedure that we're going to do. It's a theory. Okay, it's called the cell theory, and it's because we have lots of investigations and observations to support it. Number 9, in an experiment to determine the effect of exercise on pulse rate, a student checks his pulse before and after exercising for several minutes. The purpose of checking his pulse before is that it, all right, so again, it's like having 2 groups, right? We've got the group that we're doing that's not exercising, right? And we've got the group that we're doing that is. So you do take the pulse before, control, take the pulse after, experimental. You can compare the 2. So number 3 is the best answer for number 9. Number 10, an experiment is carried out to determine how different pH values of soil affect the growth of tomato plants. So again, we have a problem here, and the problem is going to tell us what we're changing, which is the pH of the soil, so how acid or base it is, and how that's going to affect the growth of the tomato plants. So we're going to measure the growth of the tomato plants. That's the dependent variable. So growth could be measured in the height of the tomato plants. So number 1 for 10. Number 11, why do scientists consider any hypothesis to be valuable? So a hypothesis requires no further investigation. That's not true, right? You can do a hypothesis and test it multiple times to see if you get the same result. A hypothesis may lead to further investigation even if it is disproved by the experiment. That is also true. So rejecting your hypothesis doesn't mean things stop. It just means that you might have to change something about your experimental design, check for possible sources of error, or just see if you can confirm that hypothesis by getting the same explanation that you did the first time. A hypothesis requires no further investigation if it is proved by the experiment. A hypothesis can be used to explain a conclusion even if it's disproved by the experiment. So the reason it's valuable is a hypothesis could lead to further investigations, whether it's supported or not. So best answer there is number 2. All right. An experiment is performed to determine the effect of watching basketball games on pulse rates. 10 students agreed to wear devices that monitor their pulse rates while watching a basketball game between competitive opponents. So like watching the NBA finals right now. Their pulse rates were measured every minute for 5 minutes in the first quarter of the game. The data collected indicated the pulse rates did not change significantly during the monitored period. State one way that this experiment could be improved to obtain a valid conclusion. So they're giving you a little bit of extra information here than you really need. We only have one group of students, okay? And we have no comparison group. So we have no idea what happens when you're not watching the competitive sport, or we don't have a group that's not watching the sport to compare back to. So that's one problem. You could say there's no control group, right? Another issue, if you wanted to identify it, would be that there's only 10 students that are in our experimental group. Excuse me. And so we would want to up the number of test subjects to make our conclusion more valid. So those are 2 possible improvements that we could make to this experiment to make it more valid. So add a control group or increase the number of test subjects. Oops. In an appropriately designed experiment, a scientist is able to test the effect of. All right, so do we want one variable? Yes or no? We want one variable. So that we want one thing to change, and that's going to be one independent variable. All right, everything else should be constant. So a scientist should be able to test the effect of just one variable. Get rid of the multiple variables. All right, a scientist is able to test the effect of the hypothesis. The hypothesis is not the effector. That's not what's happening. And is to test the effect of scientific observations. Scientific observations have no effect on anything. They're just simply an observation. So best answer there is number 1. Number 14, which statement describes the best procedure to determine if a vaccine for a disease in a certain bird species is effective? So now remember, when we're designing an effective experiment, we have to have a control group and we have to have an experimental group so that we can compare the results, okay? So vaccinating 100 birds and exposing all of them to the disease is not going to give me a control. Vaccinate 100 birds and expose only 50 to the disease. Well, we're getting closer, but we don't want to vaccinate all of them because we're testing the vaccination. So that's not going to work. Number 3, vaccinate 50 birds. Do not vaccinate 50 others. All right. So we've got two groups that meets that criteria, one vaccinated and one not, and then expose all of them to the virus. So that, my ladies and gentlemen, is the correct answer. All right. So we're going to have 100 people in our experiment. 50 of the birds, I shouldn't say people, birds. 50 birds get the vaccine, 50 birds do not. And then we expose all of the birds to the virus to see whether or not the vaccine works. So that's the best answer, number 3. Sorry. My bad, folks. Let me get back down to the next question here. All right, number 15. In one of his experiments with pea plants, Mendel noted that 75% of the offspring of a certain cross had green pea pods. That statement would be considered. So if he's noting something, that's the same as saying he's observing it. So he observed that 75% of the offspring of a cross between the yellow and the green had green pods. Okay, that's fine. So that's going to be an observation. All right. Number 16, to test the effect of hormones on plant growth, 6 potted plant seedlings of the same species were measured and then sprayed with auxin, which was a growth hormone. Okay, so what we're changing is the hormone, which is adding auxin to the plants. And then our dependent variable is to measure the size of the plant, so to measure their growth. So after 4 weeks of growth, under ideal conditions, the plants were measured again. To set up a proper control for this experiment, the investigator should. So I'm spraying some of them with the hormone, and then I need to keep some normal. And so the normal condition would be to spray with them with something that's not got the hormone in it, such as water. All right. So our best answer here is going to be number 4, grow another 6 plants, because we want to use the same number, of the same species as we started with, under the same conditions. So they have all those constants. And then we're going to spray them with water only. So we have a comparison for what happens with auxin compared to what happens with just regular water. Okay, so best answer there is number 4. Last question. Diagrams, tables, and graphs are used by scientists to design a research plan. No. Test a hypothesis. No. Organize data. Yes. So best answer there is number 3. So please take a look at this and check your answers if you've already done it. If not, you can certainly do them with me. So have a good rest of your day and or night, and I will see you soon.